This microscope, Jeff, to look at very, very small specimens, organisms. It has capabilities to magnify about 400 times normal power.
So I could see something like an ant?
Oh, you could see the small parts of an ant. An ant would probably be too big. You could see cells. You could look at small microscopic organisms.
Ah, I'm interested. Okay. I want to see that. Okay, so I assume that I need to know a few parts here. Tell me where I should start.
Let's go over the basic parts first, Jeff.
Okay.
There are two types of lenses on the microscope. These two here are called ocular lenses, and they're what we look through.
All right.
If you look at the top here, you'll see that it has a 10x on it.
10 times?
So 10 times the normal power is what these are going to magnify.
Okay.
Because everyone's eyes are set slightly different within their faces.
Mine are fairly narrow.
We can move them back and forth.
Oh, okay.
So we just pull this side to side. There's also a little scale on top there. So once you find a setting that's comfortable, if you make a mental note of that, you can automatically go back.
Let me see. Oh, I like right there.
Perfect.
I'm a 53.
Okay. The other lenses on the microscope are these lenses right here, and they are called objective lenses. And these are the lenses that allow us to view specimens at different magnifications. They are set on something called a revolving nose piece. And if you just want to turn that, you can
sort of looks like udders of a cow.
Kind of, yes.
I can turn this?
You can turn it.
All right. So there's a little one there now.
Yeah, the little one is the low power objective lens, and on its own it has a magnification of 4. So when we combine the two, you multiply the numbers, and we would have a total magnification of 40.
40. I'm good at math. 40.
Yeah, you teach math.
I do.
The
Switch
medium
again?
power lens, yeah, it has a magnification of 10, so what would the total magnification there be?
10 times 10, I'm looking at 100.
Perfect.
And is there another one?
Yeah, that's the high power, and it has a magnification of 40, so we're looking at specimens at 400 times their normal power.
But this one doesn't have anything on it. Does it have one?
This one there is a blank one, and that would be for an oil immersion lens, and that would have the capabilities to multiply magnification up to 1,000 times. That's
Perfect.
For bacteria.
Perfect. So I've got the regular one there is what, 4?
4.
And then?
10.
10. And then?
And then 40.
40.
And
okay.
you have to remember to multiply the 10 factor.
Gotcha.
Okay. Next structure to make note of would be the stage.
Stage.
And the stage is this great big black platform.
That's a good name for it, eh?
The stage.
Yeah, the slides on the stage, a performance.
Exactly.
Okay.
And we're going to see organisms performing for us briefly. In order to hold the specimen or slide in place, there's also a stage clip, and it is spring-loaded.
The stage clip, okay.
Yeah, and it holds the slide nicely in place. Something that's very interesting about the microscope compared to other optics is that it's the actual specimen itself that moves in order to focus. If we think about cameras and telescopes, usually it's lenses that move that allow us to focus. With the microscope, we move the specimen. And in order to move the specimen, we're going to use these two controls here.
A focus thing.
The focus thing. And
there's a big one and a little one.
There's a big one and a little one. They're found on both the left and right-hand side.
Okay.
The very big outside one is this one right here. And if you want to give that a spin, let's just get this back.
It won't go this way. Oh, it must be at the bottom.
Yeah, it's bottomed out.
Oh, here we go.
Yeah, you can see if you turn the coarse focus, the stage moves up rapidly.
Pretty quick, yeah.
Yeah, now try moving the fine focus.
Oh, you can hardly see it moving.
But you can barely see it move. And that's what we'll use to move the specimen in order to focus
I'm with you.
different magnifications. Another name for this compound microscope is the light microscope, and that's because we transmit light through the specimen in order to see it. And because of that, light control is very important. There's a number of different light controlling knobs or mechanisms on here. First one is the on-off switch.
Can I turn it on?
Yeah, turn it on. And you can see the light coming up through the condenser on the bottom.
I do.
Yeah. The next device is a dimmer switch on the side there. And
Dimmer.
Yeah, it goes from 0 to 10.
So if it's too bright, you can...
You can turn it down.
All right.
0 is right off, so make sure you have it somewhere within 1 to 10 to start with.
That'd be a good trick to play on somebody.
You can do that.
Put it right at 0 and they look and they couldn't see it.
Good old science fun. I suggest you start somewhere in the middle, 4 to 6.
4 to 6,
If
okay.
you start really bright, when you look through there, it hurts your eyes.
Okay.
And you're going to want to adjust the dimmer switch according to the thickness of the specimen and also how darkly it's stained.
So if it's a thicker specimen, I might need a bit more
A
light?
little bit more light. If it's darker, more light as well.
Gotcha.
The other light controlling mechanism is right underneath the stage, and it's something called the diaphragm. and you'll see that there's a little black lever there.
Oh yeah, I see it.
That little black lever, you move it back and forth, and
Yeah.
it's called the iris diaphragm. It functions very similar to the iris in our eyes in that it gets bigger or smaller, and it's the colored portion of our eye, and it allows more or less light in.
Oh, okay. So it just is sort of a switch thing. It seems like it only goes 90 degrees.
Yeah, it's just a little lever, and you move it back and forth.
Okay.
And what it does is it controls the amount of light and the amount of stray light. And when you use that device, you're going to see that you're going to have better clarity or resolution.
It seems like I'm going to have to play with it a bit.
You're going to have to play with it to figure out how it works.
So I could do a combination of this light thing and the diaphragm thing to sort of get the best looking
Yeah, and
specimen?
that's the ideal thing to do.
Perfect.
And those are the parts.
That's it.
Yeah. Are you ready to look at something, Jeff?
I am.
All right. Eric
can I
has
do?
prepared a slide for us. And in there, we're going to have
Looks
a whole
dirty.
bunch. Well, no, those are the things we're going to be looking at.
Oh, right on.
Okay. We should see some freshwater crustaceans in there, Jeff.
So, I don't know if I've ever seen a crustacean. So I pull this thing and then I
Yeah,
slide.
and you slide it in 90 degrees to
and
the
there's a
opposite
little
corner.
slot to put it in
Yeah,
there.
and then you want to gently release that back to the slide.
You don't want to sort of let it go.
No, it may shatter the glass.
Oh, not a good thing. Okay.
A couple of things that we want to always make note before we start to view a specimen is number one, that the stage is right down, and it is for us. And and we use the coarse focus
right,
to get
yeah,
it
it's
there.
down.
That's
Okay.
one thing.
Okay.
The second thing we want to make sure we always have is the objective lens on low power.
So it has to be on low.
Yeah, and this is where we always start.
Always start
and
on
it's
low.
for safety and to preserve the specimen so we don't shatter the glass.
Okay. Okay. Can I look now?
Yeah, what you can do is you can look through the oculars and then using the coarse focus knob at the same time, bring the specimen up until you can start to see something.
Gotcha. Okay, so let me see if I can see anything. I just see white. So I use this coarse focus to bring it up.
You bring it up.
I'm bringing it up. I still don't see anything, Kelly.
You will soon, Jeff.
Oh. Here we go.
Can you see anything moving yet?
I do.
And what you'll want to do is get it as focused as possible with that coarse focus.
That's an interesting, because it's really small on there, but when I'm looking in here, it looks pretty big.
It's pretty big. It's how many times bigger than normal would it be?
It would be 10 times 4.
Excellent.
40. Right on. Okay, can I go into medium?
I would maybe just play with the fine focus first of all.
Right.
And something else that you're going to want to do is, because when we go to higher powers, we're going to see a smaller amount of the specimen, or our field of view will decrease,
Right.
is that you're going to want to center your image.
Oh, okay. How do I
And
do that?
on the side here, we have got something
This
called a
little gadget?
mechanical stage adjustment, and it's really two knobs built into one. And if you turn those knobs, you'll see that one of them is going to move the specimen up and down, and the other one moves it side to side. So you'll want to pick an organism out that's moving,
Okay.
and get it nicely
Okay.
centered.
Centered. Oh, I see. So one goes left to right, and the other one up and down. Okay.
So
I'm
you're-
perfectly centered. I'm perfectly focused.
Excellent. Now we're ready to go to medium power.
I'm ready.
And you're going to take the revolving nose piece, and you're going to clip it
This is sort of exciting.
into its slot.
Oh, so it clicks in.
Yes, it clicks in. Something that you'll also notice is that the distance between the lens and the slide decreases.
Oh, I can see trouble there.
Yeah.
Like if
Trouble
I'm on
will
high
lurk
power?
ahead if you're not careful.
If I'm on high power,
We're going to see it's going to get less than a millimeter, and we don't ever want to touch the coarse focus knob now. From here on in, it's fine focus only.
so once I'm on low and I focus it, only on fine.
From here on in.
Gotcha. Oh, so it's a little off kilter, so I'm going to have to...
You're going to have to fine focus it. The microscope is parafocal, and what that means is that it's roughly focused on every power, but not perfectly, so you'll have to use that fine focusing mechanism to bring it
Gotcha.
into focus.
And then I should center things too?
I would center it again, because when we go to high power, our field of view decreases, and if we're not right centered, we may not see our specimen.
So this is 10, right? And 10.
That's 10.
So I'm looking at this 100 times.
100 times its normal power.
More than normal. I like it. I think I'm ready for high power.
You are, then revolving nose piece, and this time you're really going to want to be careful when you click it, just to see how close it gets.
Okay.
If at any time it looks like it's going to touch, then don't force it.
I think I'm okay.
Okay.
Snap.
Now you can really see why you don't want to use that coarse focus knob, because if you were to accidentally turn it the wrong way, you could shatter both the lens and the slide.
Not good.
No.
And the lens is worth...
Hundreds of dollars.
We'll say 1,000.
Okay, inflation.
Oh, I'm out of. It looks like I need to focus a bit.
And again, you're doing things right. You're using the fine focus.
Oh, very neat.
And can you see some of those little crustaceans moving around, Jeff?
Yeah, yeah. I hope I can. That's neat. It's sort of funny though with this one, like the other ones, I could see an object. And when I focused, it was perfect on that object. But in this one, it seems like I can focus on one thing, and then if I move the thing, it focuses on another thing. Why is that?
Yeah, that has something to do with what's called planes of view.
Oh,
and you're able to actually focus on a single cell layer here. And if you start to change the fine focus, you may focus on this level, or this level, or this level. And because of that, you may see different levels of organisms or different cell layers.
I see.
So you're going to constantly notice that you're going to have to be fine focusing your specimen.
Yeah.
Because the mechanical stage adjustment is on the left-hand side, I would get into the habit of fine focusing with your right hand so that your left hand is freed up to move the specimen up and down and back and forth.
Over here. Oh,
Yeah.
I get you. So I hold fine focus here, move things up or down.
Exactly.
Oh. There we go. So I can move up and down, and I focus on different, what did you call them, layers?
Yeah, there'll be different layers there, or different planes of view.
Onions have layers.
They do.
Sorry, little thing about Shrek there. Okay. That's very cool. Am I done? I like that. I could look at that for a while.
You're finished looking at the specimen, but we always want to make sure that we return the microscope back to its normal position, and we put the slides or specimens back where we got them from.
So what should I do? Like, I know I don't want to hit that lens with this thing. Should I use this thing to go down, or what should I do first?
There's a sort of a procedure that we use to ensure that we don't break anything. The first thing that you'll want to do is you'll want to turn the revolving nose piece right to low power.
Ah, gotcha.
And that'll immediately get some distance between the specimen and the objective
Well,
lenses.
there I'm on blank. Would blank work
Either
too?
or, yeah. It's nice to put it on low so that the next person that comes in doesn't have to,
Gotcha.
to worry about that.
I'll put it on low.
And now what we can do is we can take our our coarse focusing mechanism and lower it right to the bottom.
Okay, good.
And then remove the slide, and you can, in this case, you could dispose of it in the appropriate place, or if you were using a prepared slide, you would put it back into the box that you got it.
Good. And then turn it off?
Yeah, you would turn it off, and you could wrap the cord around the base and return it to where you got it, and you're finished.
That was pretty good. That's not too bad.
No, not at all.
I guess I've got to worry about my procedure though. So when I put a slide in there, I got to make sure I'm on low, focus,
For sure.
use my coarse one and my fine one,
Yeah.
and then as soon as I go to medium, no more coarse,
Normal, fine focus only.
fine focus. Go fix it on medium, get to my focus, get my thing oriented in the right spot, go to high, focus again, take a look at what's there. Very cool. Well, thanks very much,
You're very
Kelly.
welcome. That was lots of fun.
That was a great introduction to the microscope.
It was.
Okay, thanks.
Thank you.